Welcome back, everybody, to our Introduction to Chemistry lecture. This is the second half of the lecture where we're going to talk about weak chemical bonds, water, solutions, solutes, solvents, acids, bases, things like that. Believe it or not, the strongest bonds found in the molecules of organisms are covalent bonds. A lot of students sometimes find this hard to believe because the idea of an ionic bond with a positive and a negative thing attracting each other, that sounds strong, but it's actually quite a weak bond. So weak chemical bonds, such as ionic bonds and hydrogen bonds, are really important to an organism because weak chemical bonds reinforce the shape of large molecules. They help molecules adhere to one another. But the problem with a weak chemical bond is that they are transient. It means they break and reform very quickly. They don't last very long. Whereas when you form a covalent bond, unless something maybe enzymatically is going to happen to break that bond, that is a very strong bond that will last through the lifetime of the molecule. Together, multiple weak non-covalent bonds can produce highly stable structures. So we're going to take a look at the most common weak chemical bonds. Specifically, we're going to look at the hydrogen bonds, and we're going to look at van der Waals interactions. So the weak chemical bonds of ionic bonds we've done already. We're going to look at hydrogen bonds and van der Waals interactions, and those are going to be the three weak chemical bonds that we'll look at. A hydrogen bond forms when a hydrogen atom, which is covalently bonded to one electronegative atom in one molecule, is attracted to the electronegative atom in another molecule. Now, this is because in the electronegative molecule that has the hydrogen has a slight positive charge. And the slight positively charged hydrogen is going to be attracted to the slightly negative electronegative atom in the adjacent molecule. So in these electronegative atom containing molecules, for lack of a better word or lack of a better phrase, there are positively and negatively charged ends. A hydrogen bond forms between the slightly positive end of one electronegative compound and the slightly negative end of another electronegative compound. So it's not really a bond, actually, but an interaction between two dipoles, between the slightly positively charged hydrogen and a slightly negatively charged atom in another molecule. They are intermolecular bonds. They are found between molecules. They are not found as intramolecular bonds. Intramolecular bonds would be the bond between the oxygen and the hydrogen in the water molecule. Let's take some time to take a look at these hydrogen bonds in more of a, I don't know, a realistic situation. Let's talk about the hydrogen bonds that form between the polar molecules known as water. Now remember, this is a polar molecule. It's an unequal sharing. There's going to be dipoles. There's going to be the slightly positively charged end, which is the hydrogen end, and the negative end is going to be the oxygen portion of the molecule. Here we have a picture of 1, 2, 3, 4, 5 hydrogen molecules. These dotted lines, which are found in between, are going to be the transient hydrogen bond that's going to break and reform. And again, the hydrogen bond forms between the hydrogen of one molecule and the slightly negative end of another adjacent molecule. In this case, just happens to be another water molecule. So you can see these hydrogen bonds forming between the slightly positive hydrogen end of one water molecule and the slight negatively charged end of an adjacent water molecule. If electrons are distributed asymmetrically in molecules, they can result in hotspots of positive or negative charges. Van der Waals interactions are attractions between molecules that are close together as a result of these positive-negative charges. This is another type of intermolecular bond. It's found between molecules. It was actually named after the physicist Johannes Dietrich van der Waals. It's often used to describe the totality of weak intermolecular forces. And collectively, a bunch of van der Waals interactions can be quite strong. For example, the van der Waals interactions that are found between the hairs on a gecko's toe and the surface of a wall. The totality of all those van der Waals interactions allows a gecko to scale a wall. I kind of think of van der Waals interaction as being a hydrogen bond that doesn't involve hydrogens. Because again, it forms between the slightly positively charged end of one molecule and the slight negatively charged end of a neighboring molecule. Chemical bonds are really important because they determine the shape of a molecule. For example, the angle between the oxygen and its two hydrogens is a fixed angle of 104.5 degrees. And a molecule's shape is very, very important to its function. For example, molecules such as hydrogen gas or oxygen gas are always linear. But most molecules have very specific three-dimensional conformations or three-dimensional shapes. So a molecule's shape is determined by the positions of its atoms' valence orbitals. When a covalent bond forms, the valence electrons will undergo rearrangement. Specifically, it is the electrons in the s and p orbitals that will undergo rearrangements and may hybridize with each other as they shift and rearrange. And this is going to create specific molecular shapes. For example, for methane, this shifting of valence electrons so that you can form covalent bonds between the carbons and the hydrogens creates a tetrahedron of four teardrop-like orbitals that the carbon is going to form. And then those teardrop orbitals are going to interact with the s orbitals of the hydrogen to form kind of a pyramid shape. It's a pretty symmetrical shape that you'll see, and it is because of the orbital shape that the carbon atoms have combined with the orbital shape that the four hydrogen atoms have. For water, the interaction of these orbitals is going to create a V shape or an asymmetrical shape. The reason why shape is important, because biological molecules recognize and interact with each other based on molecular shapes. So let's take the example we see here in this slide, which is going to be the binding of a ligand with a receptor. The ligands in this case are going to be a naturally produced painkiller called an endorphin, or a pharmaceutically produced synthetic version of this called morphine. And both of these ligands will specifically interact with a protein called a receptor. Now, if you look at the natural endorphin and the morphine, they don't have the same overall three-dimensional shape. But if you notice, the portion that actually physically interacts with the receptor is identical. So molecules with similar shapes can often have similar biological effects. The fact that the endorphin and the morphine have the exact same shape where the ligand interacts with the receptor means that morphine is going to pretty much do the same thing that the natural endorphin does. There are very powerful computer programs that can predict the function of a molecule by looking at its three-dimensional structure. Let's move on to chemical reactions, which are the making and breaking of chemical bonds. You guys know that the starting molecules of a chemical reaction are called reactants, and the final molecules of a chemical reaction are known as products. Chemical reactions are going to occur when new bonds are formed, or where bonds are broken, or maybe when bonds are formed and bonds are broken. Now, there are three basic types of chemical reactions that are found in cells. We have the synthesis reaction, in which two reactants are combined to form a product. So that is going to form a bond. That's called an anabolic reaction. We've got our decomposition reactions, in which a bond is going to be broken and the reactant is broken up into its constituent components. This is known as a catabolic reaction. And then cells are also capable of undergoing what are called exchange reactions, in which bonds between existing reactants are broken, the reactants are combined, and new bonds are formed. That's what this slide is showing. In a synthesis reaction, bonds are formed. In decomposition reaction, bonds are broken. And in exchange reactions, existing bonds are broken and new ones are formed in the process of making products. These three types of reactions go on constantly in both prokaryotic and eukaryotic cells, and collectively we call these reactions that go on within a cell metabolism. It's important to remember that when you write out chemical reactions, they have to be balanced. This is because of the law of conservation of mass, or what you can call chemical bookkeeping. The law of conservation of mass says, hey, you can't destroy or create matter, and that means that the number of atoms of each element have got to be the same before and after a chemical reaction. Simply put, the law of conservation of mass says the number of atoms in the reactants have to equal the number of atoms in the products. But you guys are probably used to writing balanced equations because you have chemistry as a prerequisite. All chemical reactions are reversible. This means that the products of the forward reaction become the reactants for the reverse reaction. And in chemistry, equilibrium is reached when the rate of a forward reaction equals the rate of a reverse reaction. And believe it or not, in living organisms, chemical equilibrium is something you don't see very often. Let's spend some time taking a look at a very important molecule for the viability of every single organism on this planet. It's called water. So here are some properties of water that you probably are aware of already. Water is liquid at room temperature. Specifically, it's liquid between 0 and 100 degrees Celsius. That means we as human beings can drink it, but it also means that water can move in and out of living organisms very easily. The temperature of water rises and falls very slowly, and this is a property of the hydrogen bonds. They can absorb a large amount of energy before they break. So this prevents sudden and drastic changes in temperature in cells undergoing metabolic reactions. A lot of metabolic reactions are going to produce heat as a byproduct, and without the ability of water to absorb this heat, well, the temperature of the system would change dramatically. It actually requires a high level of heat to change liquid water into gaseous water. For an organism like us, we can take advantage of that in order to cool our body temperature. And here's something you may not realize, but frozen water is less dense than liquid water. We're going to see in a couple slides that when you drop the temperature of water, the adjacent water molecules are going to increase in distance. So the distance between neighboring water molecules gets bigger. And this leads to a drop in density of solid water, which is known as ice. Put it simply, ice floats. And for an aquatic organism, this is really important because water will freeze from the top down. So if you're an aquatic organism in a deep body of water, it's unlikely that that entire body of water will freeze. And all you have to do is go down to the deeper levels of that body of water where the water is still liquid. And basically, you'll hibernate through the winter with that level of ice on top of you. If you are an aquatic organism that lives in a shallow body of water, you are probably going to freeze along with the entire water in that body. Some organisms are capable of surviving that freezing, but other organisms are just going to die when the water freezes. Here are 4 of water's properties that facilitate an environment for life. And these 4 properties are water's versatility as a solvent, the cohesive behavior of water, the ability of water to moderate temperature, and the expansion of water upon freezing. Let's take a look at water being the universal solvent. Water facilitates most chemical reactions in a cell, so it's a very versatile solvent. Its polar covalent bonds and its bent shape allow for water molecules to interact with neighboring molecules very easily. And the forming of breaking of hydrogen bonds between water molecules and adjacent molecules can allow for the even distribution of dissolved substances. As a matter of fact, water is the universal solvent for both polar and ionic compounds. The definition of a solvent is a liquid that dissolves a solute. So of course, the definition of a solute is a substance that dissolves in a solvent. Water is the most abundant and most versatile solvent in the human body. It's also one of the most abundant and versatile solvents in just about any organism on this planet. But the thing is, not all solutes dissolve in water. Hydrophilic solutes, the ones that are water-loving, these will dissolve in water, whereas the hydrophobic or water-fearing solutes do not interact and dissolve in water. Now, by dissolve, what I mean is that the substance that is interacting with the water molecules interacts in such a way that it becomes completely surrounded by water molecules. This is known as a hydration shell. If you are hydrophilic, you can be surrounded on all sides by water molecules. Hydrophobic solutes do not become surrounded on all sides by water. As a matter of fact, they don't interact with water molecules at all. And a great example of this would be the interaction between an oil layer and a layer of water. You take oil and water and you shake it really, really hard, and you think that you have dissolved the oil droplets within the water. But because the water molecules do not interact with the hydrophobic oil particles, the water molecules instead are attracted to each other. And this squeezes the hydrophobic molecules out and separates the oil into a separate phase. So it actually results in what is known as phase separation. Why is water an incredibly versatile solvent? Well, this is because of its polarity. It actually allows water to form hydrogen bonds easily with other charged molecules. So let's take the example of an ionic compound such as sodium chloride. If you take sodium chloride and dissolve it in water, each ion is going to become surrounded by water molecules. Now, this is the process of dissolving when a compound becomes surrounded on all sides by water molecules. This is known as solvation, or it results in the production of a hydration shell. And the reason why it happens is that the electronegative oxygen of the water molecule interacts with the positive sodium in the salt crystals and pulls it away from the crystal and surrounds that positive sodium atom, whereas the positively charged hydrogen of the water molecule is attracted to the negative chlorine or chloride ions and pulls those away from the salt crystal and surrounds those. So that eventually all sodium and chloride ions are pulled away from the crystal lattice structure and surrounded on all sides by water. So each of those two ions, or all of those sodium and chloride ions, will be surrounded by a hydration shell. This is actually the process of dissolving or solvation. The example in the previous slide was the dissolving of an ionic compound into individual charged ions and the surrounding of those ions with water. Well, that's easy to understand, but also water can also dissolve compounds that are polar. Even large polar molecules, such as big proteins, can dissolve in water if they have ionic and polar regions that will interact with water molecules. Here we have a very large protein which is surrounded by a hydration shell of water molecules because there are regions of slight negative and positive charges in this large compound that you can see. The solubility of an uncharged substance in water depends on its ability to form hydrogen bonds with water. So the dissolving means to become surrounded by water or to have a hydration shell. A mixture is defined as a substance that forms when two or more types of elements or molecules are physically blended together in a solvent without the formation of physical bonds between them. In other words, the components of a mixture can be separated from one another. So here we have this idea of we have a mixture of different colored jelly beans. There are no physical bonds that are forming between the jelly beans. I mean, mainly because jelly beans can't form bonds. But the fact that there's nothing physically joining these jelly beans to one another means I can separate them out into their individual colored groups. That's a mixture. A substance that forms when you blend two or more things together without forming physical bonds between them. There are two types of mixtures, homogeneous mixtures and heterogeneous mixtures. In homogeneous mixtures, the composition of the mixture is uniform so that every part of the mixture will be the same. This is actually known as a solution. And it's formed when you take a single solute and you dissolve it in a solvent, so that no matter where you sample that homogeneous mixture, you will find the solvent and the solute. For example, if you took sucrose and dissolved it in water, no matter where you look in that sucrose solution, you will find sucrose in water. In contrast to a homogeneous mixture, which is known as a solution, there is also what are called heterogeneous mixtures. in which the composition is not uniform and every part of the mixture is not the same. There are two types of heterogeneous mixtures we're going to look at now, and these are known as colloids and suspensions. A solution is a homogeneous mixture, and it is made up of a single solute that is uniformly or homogeneously dispersed through a solvent. So that when you sample the solution, the composition of the sample is going to be the same no matter where you have taken the sample from. So for example, if you were to take sodium chloride and throw that into water as a solvent, you would end up with a sodium chloride solution. And if you looked anywhere in that solution, you would find sodium, chloride ions, and water molecules. Many substances release ions when they react with water, and a lot of these substances are salts. These ions dissociate within the water to produce solutions that will conduct electricity, so that these ions are referred to as electrolytes. And most electrolytes are created through the decomposition of ionic substances like salts in water. For example, you take sodium chloride, add it to water, and you get an electrolyte solution of sodium and chloride ions. That salt water can conduct electricity. Although it's important to realize that certain polar compounds can also liberate electrolytes. A colloid is a type of heterogeneous mixture, and it's a mixture made up of larger particles called dispersed phase particles. Now, dispersed phase particles of a colloid mixture are large enough such that they can be acted upon by gravity. These particles actually have the potential to settle out because of gravity. But here's why they don't. Because they will carry the same charge, and when they get close to one another as they settle out, they will repel one another so that they remain physically suspended within the solvent. For example, let me give you an example. The plasma proteins that you find within blood, the plasma proteins dissolved in the blood plasma are known as a colloid mixture. They have a overall negative charge, but they are large enough that if you took away that negative charge and they could not repel one another, that they would settle out. But because they have an overall negative charge, the minute they try and settle out and they get close to each other, they kind of bounce off each other so that they remain suspended within the blood plasma. The suspension is a heterogeneous mixture made up of even larger components. These components are solid and they are big enough and heavy enough so that they can settle out because of gravity. So the colloid has larger particles than solutions, and the suspension has larger particles than colloids. If left undisturbed, these large solid particles that are found in a suspension will settle out because of gravity to form a solid. And a great example with that is going to be the cells, like red blood cells, you find suspended within blood. Without something physically propelling them or physically keeping them suspended, they will settle out because of gravity. Remember, most chemical reactions are going to occur in a water solvent. Chemical reactions are going to depend on collisions of molecules and atoms with one another. So therefore, they are highly dependent upon the concentration of solutes in aqueous solutions. So let's remind ourselves of a couple things. This is going to have a relationship to the second lab in the course, which is a take-home lab on molarity and concentrations and ratios. A mole is defined as the number of molecules where 1 mole is 6.02 times 10 to the 23 molecules. A mole is that 6.02 times 10 to the 23 molecules in a mole is known as Avogadro's number. Now it could be 6.02 times 10 to the 23 anything to be a mole. A mole of oranges would be 6.02 times 10 to the 23 oranges. A mole of sodium is 6.02 times 10 to the 23 atoms of sodium. The unit Dalton is defined such that 6.02 times 10 to the 23 Daltons is actually 1 gram. Now, guys, I'm going to tell you it's 6.02 times 10 to the 23, not negative 23. I cannot tell you the number of students I have on exams that when I ask them to define Avogadro's number, they write 6.02 times 10 to the minus 23. Don't do that, okay? Now, the reason why I'm telling you that a mole is 6.02 times 10 to the 23 molecules or particles or numbers of something is because molarity is the number of moles of solute found in a liter of solution. To calculate the number of moles, however, most of the time you're going to need to know the molecular mass of the compound, and that's the total mass of all the atoms in a molecule. On your lab 2, I'm going to ask you all kinds of questions. I'm going to say how many grams of sodium chloride would you need to add to 250 mils to make a 2 molar solution? The first thing you're going to have to do is know how many grams of sodium chloride make up a mole. That's basically the atomic mass of sodium chloride. Then, of course, you're going to have to do all that relation stuff. I remember in university setting up that weird relations equation where all of these things were on fractions and they all canceled each other out. I can't do these questions that way, so when you go to lab number 2 and you look up the answers, you're going to see the way that I do these questions. It is not the way that the typical chemistry student learns how to calculate these things. But I will tell you right here and right now, I don't care how you get the answer, as long as you get the answer. If your answer is right and your approach to finding that answer is valid, then I have no problem with the way you work through the problems. One last thing before we leave this slide, I'm going to tell you that lab number 2 will be on your first lab exam, not on a lecture exam, but your second lab, your first and second labs, which are take home, will be on the lab exam number 1. So there are going to be metric conversion questions on your first lab exam. There will be questions asking you about molarity and ratios and percentages. So if you can do take home lab 1 and take home lab 2 pretty easily, then you are nicely prepared for that section of lab exam number 1. Being able to determine molarity, moles, the molecular weight of a particular substance, these are all kinds of questions that will be assessed on lab exam number 1. So for example, I could ask you, what is the molecular weight of 1 mole of sodium chloride? Don't worry, I'm going to give you a periodic table of elements. You don't have to know the atomic mass of each of these. So that's a very, very simple question that I could ask and a relatively easy one to answer. But I can get a little bit more complex because this next question you see is a little bit more indicative of what you would encounter when you work in a laboratory. For example, I could ask, how much sodium chloride would you need to weigh out in order to make a 250 milliliters of a 2.5 molar solution? So make sure you go to Canvas, pull up your sitting on at your desk laboratory on molarity, and make sure you work through those questions. If you want to do more of them, you can go online and you can Google things like molarity questions. Make sure that you understand how to calculate molarity, how you understand how to calculate molecular weight, how molarity, moles, and molecular weight all relate to one another. And if you know how to do that, you'll do really well on that part of Lab Exam 1. Let's move on to the second property of water, which is the cohesion of water molecules. Collectively, the hydrogen bonds can hold multiple water molecules together in a well-ordered structure, like a chain of water molecules. This attractive phenomenon between water molecules is called cohesion. It is the result of hydrogen bonds. And at any given time, many of those water molecules are going to be linked by hydrogen bonds to form that well-ordered structure. Cohesion is actually defined as the attraction between molecules that are the same. And cohesion of water molecules can actually explain the capillary action of water transport we see in plants. This is a process known as transpiration. There's a little YouTube video for you to look at. But the cohesive force between water molecules actually helps pull water up from the roots of a large plant like a tree and move it up the large stem towards the leaves. The idea is that through evaporation of water molecules from the leaf surface, transpiration, or what I call plant sweating, the loss of that water molecule causes the pulling of water molecules up from the ground, up the stem, up into the leaf to replace the one that was lost. None of this would be possible without the fact that the hydrogen bonds between water molecules basically forms a nice, well-ordered chain of water molecules that can run from the roots to the leaves of a tree. So take a look at this YouTube video and you can see what I'm talking about. Please don't confuse cohesion with adhesion. Here is a chart which simply compares cohesion to adhesion. You'll see that cohesion is the attraction between molecules that are the same. They are a result of hydrogen bonding and van der Waals attractive forces. They can produce things like water droplets. It's important to realize, though, that cohesion can also be observed in other liquids. For example, the cohesion attraction between mercury molecules can produce mercury droplets. Adhesion, in contrast, is an attraction between different molecules. So, for example, between water molecules and the sugars that you would find in the plant's cell wall. Adhesion is the result of electrostatic attraction found between positive and negatively charged atoms. Collectively, all of the water molecules that interact with each other create a cohesive force that can be known as surface tension. Surface tension is a measure of how hard it is to stretch or break the surface of a liquid. And water has an incredibly large surface tension, much greater than most liquids, so it is very hard to break. Surface tension actually is related to cohesion. If you look at the interface between water and air, you will find a thin film of well-ordered water molecules, all hydrogen bonded to each other. And this interaction creates a strong and visible film right at that interface between water and air. It produces surface tension. And that surface tension can actually be utilized by living organisms like insects, like this water strider that literally walks on water because it's light enough that it can't break the surface tension found on the very surface of the water. And again, this is because as you get close to the air interface, you will have an increasing number of water molecules that will all be bound together by hydrogen bonds. And even though these hydrogen bonds form and break and reform and break, they do so very quickly. So in essence, you have a very thin layer of well-ordered water molecules that kind of form like an invisible film. Molecules of a liquid stay close together due to cohesion or due to cohesive attraction. But if you increase their kinetic energy and get them to move fast enough, they can overcome this attraction and be released as a gas. This transformation from liquid to gas is known as evaporation. The heat evaporation is the heat a liquid must absorb for 1 gram of this liquid to be converted to gas. And water has a very high heat of vaporization due to the strength of the hydrogen bonding between adjacent water molecules. As a liquid evaporates, the remaining liquid will cool in a process called evaporative cooling. So in other words, the hottest molecules will leave as a gas, and the remaining liquid that's left behind cools. The fact is, the evaporative cooling potential of water helps stabilize temperatures that in organisms that are water, that have a lot of water like us, or, like the typical cell. And it even is a way of stabilizing temperatures in bodies of water. As we touched on earlier, water can absorb or release large amounts of heat with only a slight change in its own temperature. This is due to the hydrogen bonds and the fact that they can absorb a fair amount of energy without changing the temperature of the water. So water prevents large changes inside cells as they run their heat-producing metabolic reactions. This prevents the cells from being killed as all of these metabolic reactions run. Environmentally, outside of cells, outside us, water absorbs heat from warmer air and releases stored heat to cooler air. So it functions as a temperature moderator in terms of the environment. So water is great at stabilizing temperature. The question, of course, is what's heat and what's temperature? Let's learn those. Kinetic energy is the energy of motion. And the reason why I define this is because heat is a measure of the total amount of kinetic energy that is due to molecular motion. So heat is a measure of the total amount of kinetic energy. The higher the motion of particles, the higher the kinetic energy, the higher the heat. What temperature measures is the intensity of heat due to the average kinetic energy of molecules. The temperature on Earth is typically measured with the Celsius scale, although here in the United States we use the Fahrenheit scale. In lab number 1, you will convert between Celsius and Fahrenheit. I promise on an exam, if I have you do this, I will give you the conversion formula. But I'm Canadian, and I was raised in Canada where our temperatures were mostly in Celsius. But I was raised in Windsor, Ontario, which is right across the border from Detroit, Michigan. So we would always get the news and the weather on the American stations. So I would watch a Canadian weather station and I would get temperature in Celsius, and then I would watch an American news station and I would get the temperature in Fahrenheit. To this day, the Celsius scale means nothing to me when it comes to temperature outside. But there is a quick and easy way of converting Celsius to Fahrenheit, and that's double it and add 32. The true measure, though, of temperature is done using what is called a Kelvin scale. And at absolute zero, or 0 Kelvin, this is when all molecular motion stops. The unit of heat that is expended in a reaction is called a calorie, and it's defined as the amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. By the way, the calories, quote unquote calories, you see on food packages is actually a kilocalorie. So it actually represents 1,000 calories. So the calories to a chemist, a biologist, and a physicist might be a little different than the calories that a nutritionist would consider. If you're in Europe, though, you don't get calories, you get joules. And the joule is another unit of energy, where 1 joule is 0.0239 calories. The ability of water to stabilize temperature comes from its relatively high specific heat. Now, specific heat is defined as the amount of heat that must be absorbed or lost for 1 gram of that substance to change its temperature by 1 degree Celsius. So for water, it's the amount of heat that has to be absorbed to increase 1 gram of water by 1 degree Celsius, or it's the amount of heat that has to be lost for 1 gram of water to drop its temperature by 1 degree Celsius. And the specific heat of water is exactly 1 calorie per gram per degree Celsius. In contrast, the specific heat of ethanol is 0.6 calories per gram per Celsius. This means it's easier to change the temperature of ethanol versus water. The specific heat of iron is 10 times less than that of water, so it's even easier to change the temperature of iron. This explains why we make a lot of our cooking utensils like pots and pans out of iron. So because of the high specific heat of water, the temperature of water changes very little when it absorbs or gives off heat. Here's another way of thinking of specific heat. Think of it as how well a substance resists a temperature change. Water's high specific heat can be traced to the fact that it has a lot of hydrogen bonds. Heat must be absorbed for hydrogen bonds to break, and heat is released when hydrogen bonds reform. So the high specific heat of water minimizes temperature fluctuations so that life is permitted here on the planet. You can see here again the differences in temperatures in the summertime between the San Bernardino Valley and, San Bernardino and Riverside and the Los Angeles airport, which is located by the coast. You may not know this, but water is one of the few substances that is less dense when it solidifies than when it is a liquid. This produces ice that basically floats in water. And this is really actually quite unusual. Now, if you go below 4 degrees Celsius, water will begin to freeze. And as water begins to freeze, as it begins to approach 0 degrees Celsius and become solid, the molecules of water begin to move too slowly to break their hydrogen bonds, and the molecules become locked in place. Now, this is the same actually for any liquid that becomes a solid. As you begin to drop temperature, the molecules slow down and they basically become locked in place. But here's the thing about water. because of its hydrogen bonds, the distance between the molecules when they slow down and lock into place becomes greater than that of the liquid form. So as water cools to solid form, the water molecules move further apart. And this is unusual. Most liquids, when they cool down, the molecules come closer together and lock in place. So the fact that water molecules move away from each other and lock into place when they solidify makes solid water less dense. Ice is about 10% less dense versus the liquid form. Now, when you begin to melt that ice and return it back from solid to liquid form, we're going to break these hydrogen bonds. The molecules are going to slip closer to each other. They're going to come closer together, and those hydrogen bonds will begin to form and break and form and break, just like they do in liquid form. Now, the reason why this is important is that if ice sank, all bodies of water would eventually freeze solid, making aquatic life impossible. Between two water molecules, something unique can happen. A hydrogen atom can shift from one water molecule to the other. The hydrogen atom is going to leave its electron behind and get transferred as a proton or hydrogen ion. The molecule with the extra proton is now called a hydronium ion, H30+, although it is often represented as H+. The molecule that lost the proton is now called a hydroxide ion or a hydroxyl group, OH negative. Biologists use something called the pH scale to describe whether a solution is acidic or basic. Now, basic simply means you are the opposite of acidic. In any aqueous solution at about 25 degrees Celsius, which is, room temperature, the product of a proton and a hydroxide ion would be constant. And you can write it as the concentration of hydrogen ions times hydroxide ions is 10 to the minus 14. The pH of a solution is defined as the negative logarithm of the proton concentration in that solution. So pH is written as the negative log of the hydrogen ion concentration. For a neutral aqueous solution, the hydrogen ion concentration is 10 to the minus 7 moles in a liter of solution. So the pH ends up being the negative log of that. So the pH ends up being 7. Here's the shortcut for this. This is a long way of just saying, look, if you have a concentration of hydrogen ions that is 10 to the minus 6 moles of hydrogen in a liter of solution, the pH is the number 6. So your pH is actually the exponent value, the positive version of the exponent value. Does that make sense? The positive number of the exponent value. So the pH isn't negative 6, it's 6. Hopefully that makes sense. But that's just a shortcut way of figuring out pH. But this slide describes the actual reasoning and the mathematics behind that number. Acidic solutions have pH values that are less than 7. Basic solutions will have pH values greater than 7. And most biological fluids have pHs somewhere in the neutral range, about 6 to 8. On this scale, you can see various things in their pHs. Up at the top of the scale, the pH of 0 would be something like hydrochloric acid. pH of 0 means you have 10 to the 0 moles of hydrogen ions in a liter of solution, and 10 to the 0 is 1. So with a pH of 0, you have 1 mole of hydrogen ions per liter of solution. Down at the bottom here, you might have something like sodium hydroxide, which would be a pH of 14. That would mean you have 10 to the minus 14 hydrogen ions per liter of solution. I'm going to warn you, in exams, sometimes I will give you the concentration of hydroxide ions and I ask you the pH. And It's not that bad, actually. If I give you let's say the hydroxide ion concentration is going to be 10 to the minus 6. All you have to do to figure out the pH of that solution is to take 14 and subtract 6. So the pH of the solution is going to be 8. If I give you a hydroxide ion concentration of 10 to the minus 2, then that means the pH is going to be 14 minus 2, and it's 12. The reason why I say this is that sometimes if you're going through an exam and you see the H, you're thinking, the pH is this. So all you have to do to figure out the pH, if I give you the hydroxide ion concentration, is to, again, take the exponent and subtract that from 14.
Biological fluids like blood pH, tissue fluids, even the cytoplasm inside cells, needs to remain relatively neutral. And in order to do so, cells will produce buffers. A buffer is a chemical or compound that keeps the pH of a solution within a normal range. Another way to look at it is a buffer resists pH changes by taking up excess hydrogen or hydroxyl ions when there's an excess, and releasing these ions when there's too little. Now, the best described buffer in organisms is called the bicarbonate buffering system. Carbon dioxide and water will combine and enzymatically will form something called carbonic acid. This carbonic acid will then dissociate into a bicarbonate HCO3- ion and a hydrogen ion. And it is the presence of hydrogen ion that determines the pH of things like our blood plasma, tissue fluids, and the pH inside a cell in its cytoplasm. The bicarbonate ion is there to combine with excess hydrogen ions in order to prevent a fluid from becoming excessively acidic. And the hydrogen ion can combine with excess OH ions to prevent the excess alkalization of a fluid. So these two components are readily available to soak up either hydroxyl ions or hydrogen ions and prevent massive changes in pH.